Our next panel up is ecosystems and the environment, and I'd like to go ahead and welcome our panel presenters and our moderator, Schery Umanzor, with the University of Alaska Fairbanks, up to the stage.
All right, 30 seconds there to settle or resettle. Yeah, welcome to this next panel. I'm Schery Umanzor, and with me, Bobbi, Jordan, Angie, and there's a test there, and Alex. Yeah, just a quick reminder that the Q&A section is at the end. You just scan the QR code. I'm sure you have heard this a lot of times. Or if you don't want to scan the code, then there's a mic or someone with a mic somewhere over there. Yep. So without further ado, let's start with here, flipping the page. Okay. Hold on. There's too much light, so I cannot read here. Let me go back. Sorry, guys. Oh, Bobbi, yes. That was a hard one.
I think I'm up first. Okay, yes, I am up first. So I'm Bobbi Hudson. I'm the executive director of the Pacific Shellfish Institute. So, wow, I feel like I'm really loud. We work very closely with the shellfish producers all along the West Coast, actually across the country, but primarily along the West Coast. So today I get to talk about carrying capacity, and in the next 10 minutes, carrying capacity 101. So I want to start off by, obviously, you have seen this term because it's on my slide right now, but show of hands, who thinks they understand what carrying capacity means in aquaculture environment? A couple. I was hoping there was going to be more so I could have a joke that, like, okay, I'll just sit down. I don't need to give this talk, but apparently not. Regardless, I do look forward to talking to everyone over the next day and a half and just seeing how I see this topic agrees or disagrees with what your vision is of carrying capacity. Okay, I figured this out. Okay, so carrying capacity, it really is increasingly referenced and questioned for mariculture development projects. That's why this issue has come to my attention and I get asked to give talks like this. Last year, before this conference, I worked with the Alaska Mariculture Alliance and actually a couple of people on this panel and others to come up with fact sheets. One of them we thought was going to be about carrying capacity. That was the topic we were after, but as we dug into it, we really decided it was more of an issue of scale. That's how we thought the concept best fit mariculture development in Alaska, because up here we have 30,000 miles of coastline, and right now aquatic farms are only 2 square miles of that. So, we need to think about scale. So specific to carrying capacity, I'm going to try to keep this really simple terms. That was my goal, and if you had my abstract I submitted for this, that was absolutely my goal, not jargony. But there are 4 different approaches to measuring and assessing carrying capacity. So we have physical, production, ecological, and social. So all of these are distinctive types of carrying capacity that can be measured or assessed, and definitely of note is that social carrying capacity then informs ecological carrying capacity. But all of these things together, all 4 of them together, constitute ecosystem-based management, which is a term that if you're in natural resources and natural resource management has been around for decades. Like, it's written into federal policies of how our natural resource managers should be looking at our ecosystems. And specifically, ecosystem-based management, I mean, these are important words, but sort of overstating the concept itself, but it's an integrative approach to resource management that considers the entire ecosystem, including humans, and the elements that are integral to ecosystem function. But what does that really mean? I serve on the International Council for Exploration of the Sea, working group on ecological carrying capacity in aquaculture, and last year we published this paper in Marine Policy where we looked at 10 different nations and do their policies actually incorporate the concept of ecological carrying capacity, and are landscape analysis, which largely was a literature review looking at individual companies or nations' policies, that term ecological carrying capacity doesn't come up. But each of these nations, or most of these nations, still had the concepts embedded in their policies. So the management is following these concepts, but especially in the US and Canada of note. So ecological carrying capacity, it specifically considers the targeted species interactions with the environment and the environment's ability to support other species' use of the same spatial area. So back to concepts that many of you might be familiar with are our commercial fisheries up here, right? Like, so the most simplest way to look at this is maximum sustainable yield. So in population ecology, you know, the most common utilization of this concept is maximum sustainable yield. So with that, we can determine the maximum population size at its, or how much extraction you can have, the maximum population size for the maximum growth rate. But even above that, and I'm reading these out of order, obviously, it evaluates the potential limitations of competition, predation, and food supply so that we can have that maximum extractive pressure that does not cause unacceptable change in the ecosystem. So my group, Pacific Shellfish Institute, we've been working on research in this area for quite some time. This research predates my time at the Institute, which is more than 15 years, hard to believe, but we were looking at production on a mussel raft. So assessing production carrying capacity to inform this farm. This is mussels in Puget Sound, and they looked at chlorophyll depletion through this raft. And so in incoming tides, we'll say the upstream versus that downstream amounts of chlorophyll through this raft. So upstream, it's higher, it depleted through the middle of the raft, and then on the downstream edge, shortly after exiting the raft, it does boost up again. But that had implications for growing mussels. So the production could be changed, and they were able to recommend that simply rotating those rafts 30 to 50 degrees to increase the flow through those rafts would increase the food amount, and then the seeding density could also be increased without reducing production of those mussels if they used ropes and biodegradable cotton socks, which they now do, to more evenly distribute those mussels so they wouldn't have these hot spots of depletion. And then they also recommended that replacing the smaller mesh predator nets with a larger mesh net once once the mussels were to a size that the predators, in this case mainly fish and diving ducks, would not get to them, that changing the mesh size or cleaning those nets more often could improve the growth rate of these mussels. So this is sort of like basic production carrying capacity. So you can also model that. So these are a couple of models that I'm familiar with. Our group and I specifically have published different species and uses of these models. So these are put out by Long Environmental. There's some website up in the top right there. Yeah, top right for you guys too. So these are a combination of production and sort of ecological models. So WindShell is a growth model to establish how an animal will grow based on the food availability and the environmental condition. So that just gives you what can you predict that animal is going to grow in a particular area based on the local conditions. And then one step further than that, FARM is really a screening tool for citing a specific species in a specific area, again, based on those environmental conditions, but then it gets the feedback of how does that growth of that animal then affect the water quality in that system. And in this case, we were talking about carbon credits earlier with regards to kelp. This model will also estimate nitrogen credits, nutrient credits. You could have phosphorus as well, but specifically gives a number for nitrogen removed from the system. And this is used in some places to estimate nutrient credits. So another more complex model, for sure, is EcoPath with EcoWin. So this is an open source modeling software suite that's been used worldwide for 40 years as of this year. So this is, again, open source, so it's easily accessible, but there's a lot of data that has to go into it. So EcoPath with EcoSim, it's a way to visualize a biomass of a suite of species, and really it takes into account the mortality, the production of the species group, the food intake, and the diet composition. So how are these animals in this ecosystem related and the primary production of that system? And then it's also really gets at the ecological and the social carrying capacity because it's a stakeholder-guided process. You gather the best available data to inform the model, to build that food web, and then you can ask relevant questions of that model. So the 1, 2, and 3 that are shown there, that 1, that is the biggest part of the work, is you got to build the food web. So you get data sources to tell you what your ecosystem looks like, how are those different circles related to each other. So you need to have a population estimate for each of these things. You need to see how they relate with mortality and predation, their overall diet. And then that number 2 is, the number 1 is present state, then you look at past. And so you have to have those data sets for both periods of time, hopefully similar periods of time. And so that number 2 is the change driver, which is what's the difference between the past and the present. So then from that, knowing the past to the present, you can then make predictions for the future. So a lot goes into it, but it's a very robust tool. So that number 3 is really what the prediction of the future could be based on scenarios that you ask through a stakeholder-guided process. But key considerations of this, those ecological carrying capacity models need a tremendous amount of data. And so I can't stress that enough. People ask me about, oh, can't we do this carrying capacity model for this particular area? And so I caution people, it really takes a tremendous amount of data to do this well. I think we all know what models are. It's, garbage in can be garbage out. So you need to have good data. So unfortunately, the species population data and trends over time doesn't always exist, particularly for a broad area. So they rarely exist for all components of the ecosystem. So it's difficult to build a robust model. Furthermore, the spatially explicit data is rarely does exist in order to inform dynamic temporal models. And then finally, that detailed bathymetric and circulation data are also needed. So in examples that I've been involved with, we already had those great models at our available for use. So we could use those models that already existed and then add the ecological data on top of it. So yeah, there's a lot that goes into it. So there is some good news though. So for shellfish and their environments, more broadly, there are these feedback loops, right? So under nutrient-limited conditions, nutrient regeneration can stimulate primary production. So that provides a positive feedback. So that's something that people do get concerned about, is depletion of phytoplankton. So there can be a positive feedback. And so in most systems, primary productivity is determined by nutrient and light availability. And grazing by bivalves, of course, because I'm most interested in bivalves, strongly influences both of those factors. So filtration lowers the phytoplankton concentration and removes that fine sediment particles. This reduces the light attenuation and increases the productivity in light-limited systems, and remineralization of nutrients from wastes increases nutrient availability in systems that are nutrient-limited. And I promise that's as complex as I'll get on any of these slides. There is a citation there. I'm happy to talk to folks about it, but then just sort of to wrap it up, here in Alaska, there really is no evidence that plankton is limiting species recovery or that plankton consumption by maricultured shellfish is having any appreciable effect on any species in Alaska. So even if the strong depletion occurs within a farm, this does not imply that there are problems with carrying capacity, either production or ecological, in the system as a whole. So with that, I will end, and I guess we are doing a panel, so I will not take questions now. So thank you.
Thank you, Bobbi. Next, we have Jordan Hollarsmith with the Alaska Fisheries Science Center, and she's going to be presenting on the efficacy of various strategies for detecting harmful algal blooms and paralytic shellfish toxins. Thank you, Jordan.
Hey everyone, thank you, Schery. Hi, my name is Jordan Hollarsmith. I'm the Mariculture Research Lead for the Alaska Fisheries Science Center. And today I'm presenting work that was led by Juliana Cornett, among many other collaborators listed here, on detecting oyster toxicity risk. I simplified the title since I submitted my abstract. So as Weatherly nicely queued us up for this topic in the previous session, but harmful algal blooms of the phytoplankton genus Alexandrium can produce paralytic shellfish toxins, which accumulate in the tissue of filter feeders like oysters, and then when consumed can result in paralytic shellfish poisoning. So it's a major public health concern in Alaska, outside of the state, for subsistence, recreational, and commercial harvesters and growers of shellfish. And there's a lot of different strategies people use to either detect the presence of Alexandrium in the water column or measure toxicity levels in the oyster tissue themselves. But there hasn't been a lot of work done to directly compare these methods to see if they're giving you the same result. Like, how do they stack up in terms of determining risk, especially here in Southeast Alaska? Well, we're in Anchorage, but in Southeast Alaska. So our group, we compared 4 methods that are used to detect the presence of Alexandrium in the water column. One was using a net tow, which is where you drag a really fine meshed net through the water column. It concentrates down the phytoplankton. You take a little sample, put it in a microscope, identify and count what's there. And it's a pretty straightforward method, low cost. The problem is it's qualitative. You don't know how much water you pass the net through, so you can't say, I saw this many cells in the microscope, therefore there's this concentration in the water column. We also used a method where we took a known volume of water at known depths, concentrated it down, then took a sample out, looked at it under the microscope, same kind of thing. And the nice thing about this is it's quantitative. You know how much water you sampled, so you can take those microscope counts and have an idea of what your water column looks like. But it does require a little more sample processing than the net tow. And then we used environmental DNA, which is where you take that known volume of water, you pass it through a filter, and then you detect the fragments of DNA that are collected on that filter to see if your target species was present. It's an incredible thing, but it does require genetics lab infrastructure, so not everyone has that. And then we also compared 2 methods that test toxin levels in tissue themselves. There's the mouse bioassay, which is the FDA-approved method used by the Alaska Department of Environmental Conservation. That's all the oyster farmers. They send in their samples to Anchorage, and mouse bioassay is the test. The nice thing is it detects total PST levels. So if you caught on Miley's presentation, she referenced the different congeners. So these toxins can take slightly different forms, which presents a challenge for measuring them. But mouse bioassay does it all, but you need a mouse colony. It results in a lot of dead mice. And then we also test the enzyme-linked immunosorbent assay, which is less lab infrastructure relative to all the other ways of measuring toxin. But it detects many, but not all, of the different forms that these PSTs can take. And so if we use the mouse bioassay as our point of comparison, since that is the industry and FDA-approved standard, what we found was that 4 other methods were significantly correlated with the results from the mouse bioassay. And what I presented is the R-squared values. So if the R-squared is closer to 1, it means there's a closer correlation with the mouse values. So we found that the C-Tox ELISA kits were very effective at detecting PSTs, which was a really great thing to see. We found that Alexandrium counts from these net tows and the discrete bottle samples did significantly correlate with toxin levels, but very importantly, there were days where we saw no cells and the oysters tested above regulatory limits. There were days where we saw a lot of cells and the oysters tested below regulatory limits. So it is not a one-to-one relationship. And we also found that eDNA can readily detect Alexandrium presence even at very low concentrations. So it could be a really powerful research tool as we continue to try to understand the complex dynamics of what triggers blooms, what triggers toxicity levels, and what completes blooms in support of industry and subsistence communities. So I'd love to talk to anyone more about this. We're working on publishing this now, so hopefully we'll be able to share that paper with you soon. So thank you.
Thank you, Jordan. Our next speaker, Angie Bowers from the University of Alaska Southeast, and she will be presenting on determining site suitability and processing potential to Alaska salmon hatchery release sites for the integration of mariculture species. Thank you, Angie.
Hey everyone, my name's Angie Bowers. I'm an assistant professor in the Applied Fisheries program in Sitka. And I'm going to talk today about a project that we've been working on with private nonprofit hatchery organizations to measure site suitability at their salmon release sites. So myself and other staff and students in the program have been working on this, and we're looking at shipping supplies and equipment to these salmon hatcheries so that they can measure the abiotic factors at these sites to determine what mariculture species might work and which ones might not. This project was funded by the Alaska Blue Economy Center at UAF, and so we're super appreciative of that. It's also kind of related to a lot of the work that Dr. Umanzor has done with her site suitability kits. And for those of you that are not familiar with our hatchery program, there are about 30 hatcheries, many with multiple release sites that release about 1.6 billion salmon here in this state every year. And so in these various bays and inlets, these fish rear in salmon net pens for 3 or 4 months every spring, and they're eating millions of pounds of fish food. And so as a result of that, they're also creating these wastes, which can become a valuable resource for integrating mariculture species. And so a lot of the work that we've done so far has been looking at kelp, but this could also be other species such as sea cucumbers or shellfish or even marine worms. We've kind of thought about that a little bit. So we're hoping this kind of form of integrated multi-trophic aquaculture could be something that could be successful here in the state. So kind of why think about integrating mariculture species? A lot of these sites are already in use, and they're in use for just one part of the year. And so while they're rearing these salmon, there's a brief window where there's really nothing going on, and then the adult salmon come back and fisheries take place. But during those months when you might want to be growing kelp, there's really nothing happening in these places. And so these private nonprofit hatchery organizations, they kind of decide what happens in these terminal harvest areas. So it would be up to them whether or not they would want to kind of diversify what they're doing or potentially lease these areas to prospective farmers. I think there's a lot of room for research in this area as far as some of the benefits that could be kind of mutual. So for the rearing fish as well, there's potential that a kelp farm, say, could be a refuge for post-release. Maybe you would see increased marine survival. They're going to be cleaning the water, whatever species this is. And so there's a lot of potential benefit. There's even some talk of looking into whether or not it would increase site fidelity with these fish, that the smell of seaweed could actually help decrease straying. And so there's a lot of work to do there, but there's also a lot of promise. And then in these areas, there's already infrastructure. There's an existing workforce in some cases, unless, of course, it's leased to outside farms. The positive environmental impacts you would see could even be more than, say, just a kelp farm, because there is that bioremediation that's happening there. And then you could see an improved public perception of hatcheries as well, and maybe even more people that would be interested in working at these sites if you have these environmental benefits that go along with it. Also of note and of interest is a lot of these hatcheries are powered by renewable energy and have hydropower. Oftentimes when you would be processing kelp, they have maybe extra space because a lot of this food has been fed out. Also potentially excess electricity, depending on what's happening there. And so just what we're doing is we've sent these hatchery organizations these giant pelican cases, and first and foremost, they contain an acoustic Doppler current profiler, or an ADCP. They're able to set this in the area of their net pens to measure the speed and direction of current, and then kind of see where these nutrients might be best utilized if you are going to integrate other species. They also get oxygen loggers, salinity, and temperature loggers, which are taking measurements every 30 minutes. And then every 2 weeks, hatchery staff will go out and do nutrient sampling, measure the turbidity, and then take out a YSI, and also measure kind of those same factors at 3 different depths and keep track of that. And so they'll do that until after those salmon are released to kind of see what's happening there. Yeah, so I've been really pleased with how much interest there is in this. I think salmon hatchery operators are excited about the potential for this, and our program has been growing kelp next to salmon net pens for 5 years now, and so we've learned a lot about what not to do, and maybe some things about what to do, and it's been really great to involve students because I think using kind of these creative solutions to issues that we have in fisheries and aquaculture is great for them, it's great for industry, and they can take that with them wherever they go on to work. And so yeah, thanks to ABEC, thanks a lot to Schery Umanzor for inspiration for this project, and I'd love to chat more about this with anyone who's interested. Thanks.
Thank you, Angie. Those are super high-end toolkits. Super jealous there. Our last speaker for this panel is Alex Lafayette with NOAA Alaska Fisheries Science Center, and she's presenting on ecosystem services evaluation of kelp farms versus natural kelp beds in Kodiak, Alaska. Thank you.
Thanks, Schery. Hi, everybody. I know I'm standing between you and the last coffee break. I'm Alex Lafayette. I'm a research ecologist at NOAA based in Kodiak, and I'm a really bad panelist because I didn't tell my moderator that we're actually going to show a short clip. We're going to share on a project that we're collaborating with Alaska Ocean Farm, so owned and operated by Alf Pryor and Lexa Meyer. And then we've also had the opportunity to work with Jamie Curry, who is a cinematographer and also Alaska Sea Grant Fellow based at the regional office with Alicia Bishop. Jamie's a curious guy, and he's a really good videographer, and we were talking about the project, and he was like, "Ooh, maybe we can make a cool clip." So I'm not supposed to let the cat out of the bag, but I am going to just tell you the impetus for the project. So there's a big growing body of evidence about the ecosystem services provided by seaweed farming. And I'm specifically interested in habitat provisioning, but a lot of the data that's published is actually from tropical systems. So colleagues and people worldwide are really curious about, do seaweed farms in temperate systems, Maine, New Zealand, Alaska, do they actually provide habitat for native species? And linking it back to this morning's plenary, Dune asked, why are we getting attention in Alaska and how can we help? And I think one of the things that we may be able to help is to quantify that question. And I don't know, I'm gonna phone a friend at the tech center. Do I just press the green? Oh, I don't press the green. What do? I don't do anything. Okay, thank you. I think that aquaculture of seaweed and shellfish in Alaska is really, really exciting. So it's a developing industry, but it's a rapidly developing industry, and people are crazy about kelp in Alaska. They really see the ecosystem benefits of it. They are developing different species and diversifying. It allows fishermen and fisherwomen to have a diversification in their industry and in their workload. And then it's producing food, and it's getting food security and direct food consumption, and then also for other products, which is really pretty cool. So I'm doing a project with Alaska Ocean Farms, and they were seeing juvenile salmon around their farm, and they were seeing lots of them and lots of fish. And so I started thinking, do kelp farms provide habitat for juvenile fish or adult fish? Is this a place where they could come in and forage, or they can come in and hide on their transit up to the rivers and streams of Kodiak? So we thought, well, why don't we take a look?
Alex approached me about using my farm as a study for this project, and I'm all for it. This is a great opportunity because I'm out on the farm. I see interaction with fish and wildlife, and it would be nice if it was quantified.
The project is really looking at, do the farms provide habitat for juvenile fish, and how does that compare to natural kelp beds? So the farmers will seed out in January, and they're mainly seeding out either sugar kelp or ribbon kelp, and then they'll grow that till it's about 2 meters in length. So it's like making this huge canopy. What a great place for a fish to hang out and hide or eat. And then they harvest it and they take it completely out of the water. And so what happens if there is a fish population in the kelp farm? What happens to that fish population? And how does it compare to a natural bed?
it's difficult because you're there working and you're looking over the side, and all of a sudden, wow, there's a flash. I'm like, that was a fish. What did I just see? You know, was that a rockfish or was that a greenling? Last season, we were harvesting on a beautiful sunny day, and I'm looking down the water, and there's fry, salmon fry everywhere. And the sun's catching them, they're flashing in the light. It was great. You know, we've seen flashes of large fish, you know, probably king salmon, you know, but you only see them for a second. So it's very difficult to confirm things because fish aren't willing to sit there. But we've seen all sorts of things. There's just a lot of life.
So we sample in the farm and in a natural kelp bed, and we're using a three-pronged approach. So we do visual surveys with GoPro cameras, eDNA sampling, so collecting water and then processing that to have a look at the genetics of who might be swimming around in the farm. And then we also want to do actual collection for biodiversity. It's basically a net that's like in a tube, and you put that out and leave it in the farm, and the fish will swim into it, and then we can collect that net. I hypothesize that we may see more fish in the natural kelp bed than in the kelp farm, but we don't know. It's
going to create a baseline, and from that baseline, I hope as the research kind of comes in, and we do more of this, that we can take farming beyond just providing a food source, and that we'll get a better understanding of the impacts on the environment. And maybe we can start helping some of these struggling systems kind of come back.
I think that the partnership between aquaculture, industry, and research is critical. So it's really an industry-driven question. I think it's the most important thing. You need trust and good cooperation with the industry to really achieve success.
Thank you, Alex, and good job, Jamie. Beautiful. So that pretty much concludes the presentation section of the panel. Thank you, everyone. And we're gonna open it up for questions. The QR code is right there, or if you have a question and wanna use the mic, feel free. Here, mic.
I have a loud voice. Has there been any implications or thoughts of folks leaving lines of kelp fallow to promote more ecosystem services and retaining those forests for those forage fish and let them just do their natural cycles?
So a lot of the farms that are permitted, they need to remove all their growing lines as part of their permit process. So to take them out either for it was in their permit application, or there may be another user group that uses it, like recreational fishing. And so I don't really, at least in Kodiak, that it hasn't been looked at, and I don't know about in other parts.
Yeah, I can give a shout out to the native village of Eak. If you want to talk to Caitlin more about that, they're trying that out up in Cordova, and there are challenges with fouling, and this is an extremely seasonal place, but something that they're looking at.
More questions before I read from the screen? No, okay. So here's a question. Do Alaska Shellfish Farm test for Vibrio, norovirus, or hepatitis A? Maybe that's a Jordan question.
This is the best question for the panel that came before us.
Yeah.
But I believe, and please correct me, but Vibrio is more triggered by water temperature and is not necessarily a regular test. And I have not heard of those other things being tested for in Alaska.
I can address that. So norovirus and hepatitis A are sort of managed under other proxies. So there's risk assessments for different species, but it's fecal coliform is the indicator for testing for those. There are tests and regulations in other countries that do require specifically norovirus testing, but that is not done in the U.S.
Thank you, Bobbi. More questions? Know that I don't have any prep questions, so please do ask. There's one in the back. Awesome.
Hi, my name is Debbie Hart. I'm a coordinator for the Southeast Alaska Fish Habitat Partnership, and we're really interested in, the value of kelps to habitat. So I really like this session, and I'm curious. I also get an opportunity to be on the Docks and Harbors Board in Juneau, and as many of you know, our harbors are very polluted often. Has anybody working on using kelps to extract pollution from certain places that might be another contributor to the industry in needing the seed and could be a part of the puzzle?
So I haven't heard that as much being talked about in Alaska, but in other parts of the country and in the world, there's a lot of interest in the power of shellfish shellfish and seaweed of all sorts as bioremediation tools, drying down excess nutrients, those sorts of things. But not as much part of the conversation in Alaska that at least I've been privy to.
Thank you. Maybe this is a follow-up question there. There seems to be a number of tools to calculate environmental services. Do you see any prospect of these tools being utilized to create revenue for farmers?
So I think it's pretty early days in Alaska. I think a lot of us know that on the eastern seaboard, there's a lot of nitrogen crediting for ecosystem services. So oyster farmers can get paid for removing nitrogen. We actually were just having a conversation about that herring will have their roe on the kelp. And so that's to a loss for the farmer, but it's a benefit for the environment. And so maybe in time, we can kind of flip that coin on its head and think about that as an ecosystem service or a credit. But I think it's pretty early days in Alaska right now in that conversation. But Bobbi,
Yeah, and I just would reiterate, I showed the farm model can calculate nitrogen credits. And those are used in like the Chesapeake to calculate, but I think the limiting factor is you have to have a market for those credits. So for farmers to actually get paid, there has to be a market, and I see very little evidence that that's happening. But that for that farm model, it is used in other countries, and it is embedded into some policies in some countries. So maybe the needle will be moved eventually.
Yeah, thank you. There's a couple of questions from the audience.
First?
Yeah, you can go first.
Thanks everyone. It's great presentations. Bobbi, I'm curious what you think kind of the biggest missing data gaps are for more of an ecosystem-based assessment of mariculture in Alaska.
I hesitate to even answer that, just that I'm not sure that it's necessary. Like if an ecosystem-based assessment is driven by concerns about carrying capacity, I don't think that we should be concerned about that. And to answer your question, I tried to stress that in a couple of slides. It's like, it's really about data. It's great to want to do these calculations, but do we really have the data available spatially for how each of these animals interact with each other, and do we have the data over time to be able to drive a quality model that will give us answers that we can believe in?
There was a question, yeah, in the back.
Hi, my name's Calvin, student at Prince William Sound College from Port Graham. I read an article that had like an isolated study of algae blooms, and one of the primary causes was runoffs. And a lot of the places that have algae blooms that have PSP are close to populated areas. And so in the study, what they did was they built little barriers in some of the water supply to the ponds that they were studying. It was a freshwater study. And it reduced the amount of algae bloom by putting up little natural barriers to filter. So my question is, do you think something like that could be tested?
Yeah, so the harmful algal blooms covers a lot of different species and a lot of different dynamics. And I think we all hear about like these harmful algal blooms with, like, I think cyanobacteria in the Great Lakes that can make it so certain communities can't drink that fresh water and that kind of thing. And a lot of those are related to terrestrial runoff and this kind of imbalance of different types of nutrients. Harmful algal blooms like Alexandrium, Pseudonychia, the kind of ones that we see in Alaska waters, are not following those dynamics. So they are just natural species in the phytoplankton community in Alaska. We don't quite know what triggers the blooms of those specific species yet. Folks like John Harley, who I don't think is here today, but he's doing a lot of work on that. The Southeast Alaska Tribal Ocean Research Consortium's doing a lot of work on that. You know, we're dipping our toes into it a little bit, but it is really important to know that these blooms can happen very far away from any human population centers, and they are not related to human-caused runoff. So it can be tempting to think, I'm in the middle of nowhere, my farm's in the middle of nowhere, my harvest site's in the middle of nowhere, so the water is clean. And yes, the water is clean, but in this clean water, and just a natural part of the system, are organisms like Alexandrium that can cause harm to us. And we are seeing perhaps different dynamics with these blooms with climate change. So there are these larger global processes that can change maybe the location, the duration, the frequency of these blooms. But it seems more related to these bigger processes like salinity input and temperature and warm water events, rather than point source pollution like runoff from a sewage treatment plant or something like that. But it's a really important question.
Right, I have two questions here on the screen that maybe are related, and Angie, I'm gonna throw them to you. Give them to you. What is the proximity of kelp farms to the net pens when they're paired together? Directly overlapping areas or nearby?
So I think this just kind of depends on the site and what is desired by the aquaculture organization. The nutrients can be pretty far-reaching, but you also don't want to be right in their way because they're driving boats and doing things around that area. So it probably kind of depends on current, also what space is available for kelp. So it'll be kind of a site-by-site there. And I think we'll learn a lot more once we get the results from the work being done right now as far as nutrient concentrations where they're sampling too.
Right. And maybe you can address this one too. Any toxicity on kelp concerns?
I think not in a setup like this. Essentially what the nutrients that they would be uptaking, these inorganic nutrients, would be, similar to fertilizing a garden. So as long as there's nothing else in the area that's negative, I would think it'd be fine.
And that question might also be referring to PSTs, the paralytic shellfish toxins, and a really important dynamic of that system is the toxin has to accumulate. And so by oysters filtering out the water, they are bioaccumulating that toxin and it builds up to unsafe levels. Whereas if it's, say, maybe a film of Alexandrium on a kelp blade caught by the mucus, I doubt there would be enough of any kind of toxin to cause concern.
Yeah. Thank you. Bobbi, this one's for you. Can you please say more about how social carrying capacity informs ecological carrying capacity?
That's a great question because I did gloss over that. So there's a lot of different ways to measure social carrying capacity, but in the examples that I gave, the social carrying capacity driving ecological is more about what questions you want to ask. So what parts of the ecosystem are the stakeholders most concerned about, so that you make sure you have the most robust data surrounding that aspect. And then for the asking questions, say in EcoPath with EcoSim, that would drive what kinds of scenarios you want to ask. So the stakeholder, there's thousands of different scenarios you could ask to try to figure out what are the relationships of things and what might happen if you change one thing or expand one thing or reduce one thing. So that's really how it works, is having the stakeholders tell you what they're most interested in so you can focus on those areas.
Thank you. And here is the last and most trending question for this panel. How best can the mariculture industry, hatcheries, and fishing industries work together to enhance the value of Alaskan seafood? And I will open up to anyone who wants to answer that. It's a great question.
I'll start. I think there's a lot of opportunity to collaborate and utilize, like, for instance, what we're looking at, this area that's only utilized for part of the year that could be farmed, but also the vessels and the infrastructure that are there, these facilities with excess power, you know, sharing processing areas, and I think just collaboration in all these areas will benefit everyone.
All right on time, so we'll wrap it up here. Thanks very much to everyone.